Yeah, good morning, everyone. I, Dr. Sakshi Bhardwaj, on behalf of Feltham Technologies, I'm pleased to welcome you all. We all are excited to have you here today. We all understand the value of innovation, science, and technologies in our daily lives, right? And how the use of computational tools has fundamentally changed development, particularly in the area of drug designing. These methods are becoming more and more popular. You know why? Because these are quicker, faster, and cheaper, as well as in comparison with the conventional ones. To get an idea how these computational approaches are integrated with the chemistry and the biology, and how these could be helpful to accelerate our research, we have our today's session, which is entitled as Basic of Computational Drug Designing and QSAR, Bridging the Gap Between the Molecule and the Therapeutics. To understand more about computational drug design and quantitative structure activity relationship, that is the QSAR modeling, we have our speaker of the day, Dr. A.R. Mahesh. He is currently working as an assistant professor at the Department of Pharmaceutical Chemistry, Ramaiah University of Applied Sciences, Bangalore. He is having over 10 years of teaching experience. He graduated from the Dayanand Sagar University, Bangalore, and done his master's and doctorate from RGHS, Bangalore. He also obtained an MBA in Pharma Business Management from ICFAI Tripura. He has earned more than 35 publications to his credential. His passion for research has led him to secure a grant as well in the area of synthetic chemistry of novel anti-cancer drugs. He was awarded Grade A for the continuous education program conducted by IISc Bangalore and has acquired proficiency in 1D and 2D NMR spectroscopy and their application, of course, in the structural biology. Additionally, he has developed an interest in the IT related application of drug designing, particularly in the molecular modeling area of the new entities. He has guided many graduates, undergraduates, and the postgraduate students. Furthermore, he has been involved in guiding research scholars in the development of antiviral drugs during the COVID-19 pandemic. He has been designated as a trainer in the faculty development program sponsored by the RGHS and APTI. He is also a member of many professional committees. Without any further ado, let's start our session. I will request my viewers to take some question and listen this session carefully, because we are going to take the question and answer at the last of our session. And feel free to drop your any queries into the chat box. So over to you, Dr. Mahesh.
Thank you very much, ma'am, for your kind introduction of mine. So, good morning, everybody. So, I am Dr. Mahesh, and I'll be, as ma'am has given you a brief introduction of myself, I'll be discussing about the basics of the computational drug design and QSAR as the bridging gap between the molecules and therapeutics today in this particular session. So, before I start my session, I just need to talk about the institute where I'm working. I am working in Faculty of Pharmacy, MS Ramaiah University of Applied Sciences. So we are 65th rank in NARF, the recently which has been declared, and our institution is fifth in Karnataka and first in Bangalore. And we are celebrating the 31 years of excellence and legacy this particular year. So this is the institution where I am working in. So acknowledging my institution, I would be starting my presentation today. So before I start my presentation, a small disclaimer is like this presentation is solely intended for educational purposes only and should not be considered a substitute for independent professional judgment. The statements of fact and options expressed in this presentation are the personal views of the participant, that is me, and do not necessarily reflect the opinion or position of my organization unless explicitly stated otherwise. All rights to the contents presented in this presentation belong to their respective owners. I have cited the respective owners then and there. Certain places have been taken from the public sources, which so with this disclaimer, I would like to start my presentation today. I would take you over from the introduction of what is a drug till and the journey of the drug design and development in this particular presentation. So coming to the basics of what a drug is, this particular definition we would have studied or read in various places. The term drug means any particle intended for the use in diagnosis, cure, treatment, or prevention of disease in man or other animals. A drug is any chemical or biological substance which is synthetic or non-synthetic. So why I'm telling you about the drug over here is when we have to go into the drug design and development, so we have to understand what exactly is a drug. So that is to be known. So that is the reason why I have just put forth what the drug is now. So the various sources of drug, if we are looking into, we have various sources of drug. Generally, the natural sources of drug include animal source. Animal source, as we know, the insulin has been extracted from the pig or cow. The growth hormone is also being taken from a man. There are plant sources, examples have been given over here as digitalis from digitalis purpurea and morphine from Papaver somniferum. There are various inorganic sources of drug like arsenic, mercury, lithium, various metals and non-metals, and also the synthetic uh, sources of drugs are chemical proper chemical sources. Like example is propranolol, biological example or synthetic compound from biological source over here is like penicillin, and from the what I can say there are various biotechnological sources or so on for the source as a source of drug. So these are the various sources of drug what has been available on the present day. So when we look into we are why are the new drugs needed in drug discovery and development, we are looking into drug discovery and development now. So you have to know why are the new drugs required on the present day. There are various unmet medical needs. There are various new diseases which were not being known earlier, which are being discovered. The diseases are being discovered now. So when the new diseases are being discovered, so we'll have to also concentrate on how to come out of this particular disease. What are the drugs or the chemical substances used to treat this particular disease or prevent this particular diseases? All those things come under the drug discovery and development. So to meet the unmet medical needs, we need a new drug, new chemical molecule is to be discovered. Next is the cost of the therapy. The cost of when you look into the Alzheimer's disease or spinal injury or cancer or all such particular diseases, the cost of the therapy will be very very high, which a normal what I can say citizen cannot be able to bear so much of expenses. So we need an economical drug for the treatment of various diseases. So for that we need new drugs to be developed. Next is sustainability or to sustain the industrial activity. Pharmaceutical industry employs thousands of people and makes massive contribution to overseas earnings, patents expiries, competition between the pharmaceutical industries. All these are the few, I mean, I can say few reasons why new drugs are being required. So now coming to the drug discovery and development. So in this particular slide, I have given you the what I can say the path to drug discovery and development. Initially, to discover a drug, the traditional way or the rational way of discovering the drug is on the present day. We do not just haphazardly or serendipitously get a drug. even though like most of the drugs are being discovered serendipitous, over here, when it comes to the rational drug designing method, we have to have a proper protocol in designing a drug or developing a drug. So initially what we do is like we identify the target. Target over here is like we identify the disease. Which disease is to be targeted, or like for what particular disease you need to develop the drug. So for the target identification is initially being done. So after the target is being identified, whether you have to what I can say like activate the target or deactivate the target, so that is being done over here by lead discovery. Lead discovery is being done the target to either activate or deactivate that particular target. So later comes like how that particular lead molecule will react with or like interact with that particular target, disease target. Then, if there are any modifications to be done on that particular chemical component, that is being done to get a better chemical component or a drug entity. After we get this one, then we go for in vitro studies. Then we go for in vivo studies where we study on the animals and so on. So then after in vivo studies, we'll be filing an what I can say like NDA. Then we go for the human trials. After the human trials, the investigational new drug or will become an approved new drug. So this is the general process of drug discovery and development. So now coming to where these computational techniques in the drug discovery are being involved. So you can see like in this particular slide, I have mentioned in each of the steps, like from target identification till the clinical trials or the what I can say the drug is being approved, computational techniques are being involved. So what kind of computational techniques are being involved is like for target identification we have proteomics, genomics, bioinformatic analysis, reverse docking, de novo designing, and so on. So next comes the lead discovery. For lead discovery, we have the computational techniques like virtual screening, focused library design, the drug likeliness studies, like for example, Lipton-Key's rule of five and all those things we would have studied. Then in then we come to like molecular docking, molecular dynamic stimulations, QSAR studies, and so on. For in vitro studies also can be done to certain extent or in computational or in silico way. That is preclinical studies can be done. The pharmacokinetic analysis can be done through through this computational techniques and simulation analysis also can be done. So coming to in vivo studies, in vivo cell culture analysis, animal model analysis, patient analysis, all these things we require statistics in it. The number of what I can say like the subjects what you take for the study would be large over here. So when the subjects are large, then you require computers to simplify our work by using various statistical methods. We analyze the particular what I can say like we come to a proper reports or like results regarding the activity of that particular drug. Similarly, when it comes to the clinical trials also, so in backhand, the patient analysis and statistical analysis is being done, which requires computational techniques. So in brief, I have just informed you, I mean, I have just showed you like how the computational techniques are being involved in this drug discovery method. So in this particular presentation, I would be mainly concentrating on like molecular docking, MD simulations, and QSAR studies. So how this molecular docking, MD simulations, and QSAR studies or like medicinal chemistry related computational techniques or chem informatics or such is being involved in the drug discovery and development. So that is the main focus of this particular presentation. So now coming to the complexity of the drug discovery, it is not so easy to come or like to discover a particular drug. So to just like when you look into like the life cycle, general life cycle or human life cycle, to achieve success in our life, we have to cross various barriers. Even over here, the drug which has to be successful should cross various barriers over here. So as we know, like no more than 10,000 drug candidates, or like just 10,000 is a number. It might go up to 1 lakh or 2 lakh or 3 lakh drug candidates. So all these drug candidates are being tested over the time to get one drug molecule. So a few of those hindrances or like the hurdles what the drug candidates undergo is like whether the drug candidate is targeted, whether the drug candidate targets the particular protein or like the targets the particular disease protein or like enzyme. Is it selective enough? Is the drug candidate or like the chemicals potent enough? Whether the drug candidate is physically stable. If the drug candidate is very much active, it is targeted, selective, but if it is not physically stable in the environmental conditions, it may get deteriorated within a day or so, then that cannot be used as a drug at all. Because like transportation requires time. The what I can say like the manufacturing to the delivery of the drug to the patient requires a lot of time. So we should see to it that the drug is stable enough. Next is like the solubility. Solubility plays a very important role in the drug absorption a- and what I can say like the binding and also the action of the drug. So whether the drug is like soluble, whether it is what I can say like perishable, or like whether it is metabolically stable. Whether it is metabolically stable in the sense like once the drug has been administered, if it gets completely metabolized into some what I can say metabolized into some metabolic products which are of non-teratogenic or like it should not be toxic in nature. It should be non-mutagenic. So it should be non-mutagenic in the sense like today you discover certain drug from the biological source or so. If the particular drug gets mutated or it mutates the proteins what it is getting binding to, then it might be very much troublesome for us. Whether the drug is patentable. Patentable in the sense, is it innovative enough? Is it innovative enough means like how is it different from the other drugs which have been present or like which is in the market or so, and whether the drug is manufacturable. After doing all the computational studies and you get one particular molecule which is not which is not been able to synthesize, which we are not able to synthesize, then that particular drug is of waste. So even if you are synthesizing, the reproducibility in synthesis of that particular drug should be sustainable. So all these things are being considered, and with all these targets or like what I can say like hindrances, the more drug candidates when it crosses all these hindrances, we may get one successful drug. So this is about the complexity of the drug discovery. So it is not that easy to get one particular drug, but to make this particular life of a drug discovery easier, we use computational techniques. So without computational techniques, it would be this one would be a nightmare for us to get one particular new drug molecule. But the computational studies have made this particular process a little bit easier for us today. So coming to the computational drug discovery, or like the CAD or computational chemistry, whatever you say, it is a chemistry whose major goals are to create efficient mathematical approximations and computer programs that calculate the properties of future drug molecules and thus helping us in process of the drug design and discovery. So here mainly what are we doing? We are taking the properties of how does these properties relate with that of the biological activity? How does this physicochemical properties of a drug molecule required for the binding of a drug with a protein? All these mathematical approximations or assumptions is being made by the computer programs, which will make our life little easier over here in drug discovery. So why computer aided drug discovery is like because of the enormous cost or increased cost and enormous amount of time. The if as we have seen in the previous slides, like more than 10,000 candidates are drug candidates are to be synthesized over here to get one single substance which is being developed. So how much cost is being involved, how much effort is being involved, how much cost is being involved, how much of money is being involved, how much of time is being involved, how much of effort, how much of labor, and all these things, how much of chemicals, all these things over here are being considered, or to make all these things a little easier to face these major challenges, we require CAD, computer aided drug discovery. So coming to the in silico drug designing, we also call this computational chemistry is like in silico drug designing, because we are using a computer or via computational simulations we are doing this particular studies, and hence we are calling it as in silico drug designing. In silico drug designing is defined as the identification of target molecule by employing the bioinformatics tools. So various bioinformatic tools are being used over here to carry out this particular study. So coming to the computer aided drug design, there are various types of computational drug design over here. So like for example, if you have known ligands and the protein structure is being known. So actually over here I have certain data over here which has been hidden. Okay, fine, I'll explain it. So there are known ligands, that means the chemical components of the drug candidates what we consider are known. The structure of the ligand is being known, the structure of the protein is being known. Then what we it is easy for us. So what we do is like we look into the protein ligand interactions. How does the ligand interact with that particular protein, and then we can go for the further studies of the particular what I can say, like discovery of drug and so on, or modification of the chemical components and so on. If in case there is no unknown ligand, so what we do is like we study the chemical properties or like the physical properties, physicochemical properties of that particular protein, which is the what is like what I can say, like which is the active pocket in that particular protein, what is the dimensions of that particular pocket, what are the now types of bonds in that particular pocket. All these things is being analyzed, and we design the ligand in that particular way. Okay, by that particular way we design the ligand, and we have a scope of designing the chemical component or the ligand or the drug candidate over here, where there we have no ligand, but we know only the protein structure. So now when you have known ligands, we have a set of chemical components which have been discovered already, and we don't know which is the target, what is this particular compound is going and acting with. But consider, you know, these kind of chemical components show the biological activity. So in this particular case, we have a scope of doing the QSAR studies, quantitative structure activity relationships. So this one I'll explain you in detail when we go to the further slides. The next part over here is like when there is no ligand known or no protein structure which is being known. So there is no protein structure. I don't know which particular protein is responsible for that but causing that particular disease. Neither I have any chemical component for what I can say, like testing that particular disease or which show the biological activity for that particular ailment. In that case, we do not have any what I can say sources for computer aided drug designing or scope for computer aided drug designing. Only thing what we need to do is like until you get a lead, you need to keep on like searching for the chemical components and going for the biological activity. So various in vitro, in vivo activities of the chemical components is the only way. When you don't know the ligand or the ligand, then we can go into the computational studies and do the studies of the chemical components. and get a better molecules for the treatment. So this is about the basics of the computer aided drug design. So next, coming to the de novo drug designing. So de novo drug designing over here comes like uh, the designing of a drug from scratch. De novo over here means start afresh from the beginning. It is a process in which the 3D structure of the receptor or the protein is being designed, or like it is being used to design a newer molecule. So this generally comes when you know the what I can say like the protein structure, and you don't know the ligand structures, or like you don't have any ligands for that. It involves structural determination of the lead target complexes and lead modifications using molecular modeling tools, and so on. Information available about the target receptor, but no existing leads, as I told you, that can interact. So we have the target protein or like the target structure, and we have to design a chemical component which actually goes and binds with this particular target. So that comes under this de novo drug designing. The general software which are being used over here, as I know, is like Grow, Legend, and LUDI. So these are the certain softwares which are being used for the de novo drug design. So coming to the pharmacophore mapping. So pharmacophore over here means like which biological activity which is being carried out, or like the part of the chemical component which actually is involved in the showing the biological activity. Pharmacophore over here is a group on a molecule or a part of a molecule that interacts with the receptor, which binds with the receptor and is responsible for the biological activity. So collectively, whole of the things is called as pharmacophore. So over here, as in a brief words, what I can say is like pharmacophore is like pharmacophore modeling is like identifying which is the particular part of the molecule required for showing the activity, required for interacting with the receptor and showing the biological activity. So for designing this pharmacophore, or like for pharmacophore modeling, we use softwares like Ligand Scout, PharmaIt, and PharmaGest. So these are the general free. I mean, I'm talking about the freewares over here. Certain softwares are freeware. Certain softwares are. I mean, we can share it, or like certain softwares have to be purchased too, or they may be available for academics for free for certain period of time. So next comes the quantitative structure activity relationships. So quantitative structure activity relationships are the mathematical relationships. So over here, what we do is like we derive an equation. Quantitative structure activity relationship is like what I can say like we have a list of chemical structures or chemical components. We know the biological activities of those chemical components. So now what we do is like we actually calculate the physicochemical properties of all these chemical components or like the lead compounds. We relate, or like we derive a mathematical equation with the known physicochemical properties with that of the biological activity of that particular molecule. Ultimately, the conclusion of this particular quantitative activity structure activity is that the biological activity of a molecule depends upon X, Y, Z properties of that particular chemical component. So further, when you are designing a new molecule, you see to it that these physicochemical properties are being retained in that particular newly designed compounds, so that the activity is being shown, or to improve the activity of the chemical component, whatever properties are being retained, those properties are being shown or to improve the activity of the chemical component are being shown. Similarly, just not that one, we are also looking into like the toxicity aspects too, which I'll be explaining in the further slide. Okay, so the softwares which we use over here is like Build QSAR and QSAR INS. So these are the softwares which are freely available, which you can use it for your QSAR study. So the next component of the computer aided designing is like virtual screening. Virtual screening is a computational filter a set of structure using one or two or more computational procedures. So here it can be used to help to decide which compounds to screen. What is the chemical compounds, or like which particular chemical molecule is to be screened, which libraries to synthesize, whether you might have like designed around like thousands of chemical components, which particular chemical compound you actually need to synthesize is being analyzed over here. Which compounds to purchase from the external company, with which the company would have what I can say spent a lot of time to synthesize that particular molecule, and in such cases what we can do is like we can easily go purchase that particular chemical component, and further you can do the research with that particular chemical component. So these are like to analyze the results of an experiment, such as like a hit to run, HDS run, and so on. So these are certain virtual screening methods over here. So next comes the molecular docking. Molecular docking over here is like identification of a low energy binding modes of a small molecule or a ligand within the active site of a macromolecule or receptor, what we call it as. Here, molecular docking is like, in simpler words, what I can say is like we have small molecules. Small molecules over here is like the ligands or the chemical components or chemical leads, what you call it as. So these chemical leads in proteins are generally big. So in which particular place, or like the active site, whether if this particular ligand goes binds, how does it bind? What are the various interactions between the ligand and the protein molecule, the small ligand and the large protein molecule? How does this, what I can say, like ligand go and bind? What is the binding affinity of this particular molecule, protein, or like the ligand with that of that particular protein? Gives you the molecular docking. This is what is called as molecular docking. How well the molecule or the ligand goes and fits into the active pocket of the protein molecule is what is this molecular docking all about. Here, given a protein and a ligand, find out the binding free energy of a complex formed by docking the ligand with that of the protein. The way it works is like Schrodinger is a paid software. BioWire Discovery Studio allows us to visualize the various aspects of molecular docking in a very simpler way, which I generally use. We have Gold and various other softwares for molecular docking. So this is about the molecular docking, and next comes the molecular dynamics. It's okay. In silico study, sorry. In silico ADMET prediction, or like what I can say like pharmacokinetic studies of the particular drugs can be done by using various softwares or like the online platforms like prediction studies. We are going to check out the absorption, distribution, metabolism, excretion, and toxicity properties of the drugs. So this is what we are going to do. Next comes the molecular dynamic simulations. So generally we do molecular dynamic simulations after we do the molecular docking. Consider the chemical component or like the ligand. We have got a wonderful binding affinity with that of protein. So molecular docking studies is being done in an environment. We remove all the environmental conditions. We remove all the what I can say unwanted substances around it, and then we check out how the molecule goes and binds with that of the drug. Sorry, that of the protein molecule. So now, when it comes to molecular dynamic simulations, what we do is like the after the binding of the drug molecule or the chemical component with that of the protein, the complex of this protein and the drug molecule, we go for molecular dynamic study. So over here, what we do is like we check the property of how well this ligand is being bound to that particular protein in the environment of the body. So we add the various salts over here, waters over here, we create an environment of the living cell, and we check how the particular what I can say the drug molecule will bind with that particular protein in the what I can say like living environment. So this environment is being created and we check. So during this particular environment, what might happen is like after the binding, because of the vibrations, because of the external waters, external salts, this particular ligand can leave out the protein. So in that particular case, the activity of that particular molecule might not be proper, and hence in the molecule there will be various validated over here by this molecular dynamic studies. So this is about the molecular dynamic study. The various softwares which are being used for molecular dynamic study is like Gromax, Desmond, Amber, Lamps, and NAMD, and so on. So these are certain softwares which are being used for molecular dynamic study. So here basic binding mechanism. How does the what I can say ligand bind with the protein molecule? Is like complementaries between the ligand and the binding site. The steric complementaries, the shape of the ligand. What is the shape of the ligand? And the site of the mirrored binding shape. How does it go and bind? So that is being checked over here. And various physicochemical properties of that particular molecule also is important over here in analyzing the what I can say like the binding mechanism. So there are various types of docking. So the various types of docking include the rigid lock and key model. Rigid lock and key model, just like in the chapter of enzymes in biochemistry we would have studied, like the internal geometry, a proper lock, a proper key, sorry, a proper key is to be there to open a particular lock. So the shape of the the ligand should properly fit into the that particular shape of the protein molecule. So that is like rigid docking or like the lock and key. Molecule is allowed to rotate, and which particular force that particular molecule will be fitting into the particular protein in a proper way. That means which particular force of the small molecule you will be getting the maximum binding affinity is being analyzed over here. Every rotation the energy is being calculated. Consider that this is the protein. Over here I have the ligand. I'll check out whether it can be fitting this like this in this way or this way or this way. All the kinds of rotations is being done, and every rotation of the molecule, the energy is being calculated. Later, the optimum force in which minimum energy is being considered as the best force. So this is about the flexible docking or induced fit theory. The types of docking include protein-protein docking, where two big molecules, two protein molecules are being taken, and there is been protein docking is being done. Both molecules over here are rigid. Interactions produces no change in the conformation. There is no change in the conformation of both the proteins. Protein ligand docking is like small molecule. The small chemical component is being docked with the big protein. Ligand over here, the small chemical molecule is flexible, but receptor or the protein molecule is rigid. Interaction over here produces conformational changes on the ligand. The small ligand, which is there, which it forms the, or it produces the conformational changes over here. So this is about the protein-protein docking and protein ligand docking. So coming to protein, I'll show you over here like computational modeling of like quaternary structure of the complexes formed by two or more interacting biological macromolecules. So there are two macromolecules in this slide. I have shown you like A and B are two big macromolecules. So when these two big macromolecules are allowed to dock, how what is the interaction between these two molecules? Protein complexes are most commonly attempted targets of such modeling, followed by protein nucleic acid complexes. So just not protein docking, protein nucleic acid complexes are also attempted targets. As you can see in this particular slide, the ligand we have a small molecule and protein is a big molecule. So there will be certain pockets in which the small molecule goes and binds with that of the protein. So or like the receptor. How does this particular ligand go and bind with that particular receptor? In which particular site of the protein does this ligand go and bind? What is the binding affinity of this particular protein? Sorry, the ligand with that of the protein. What kind of bonds are being formed between the ligand and the protein? How many number of hydrogen bonds are being present? Or like hydrogen bonds are being formed over here between the ligand and protein? Where exactly the ligand is going and binding with that of the receptor? All these aspects to be considered or to be studied over here to analyze the property of that particular ligand with that particular protein. So this is what shows us like the how the ligand behaves with that particular protein initially. So later in the molecular dynamic study, we can get to know like how well that does the ligand fit with the protein in the real time environment. So now what is this mol docking or like molecular docking score and all those things to place the ligand into the binding site of the receptor in the manners appropriate for the optimal interactions with the receptor. So how does the ligand interact with the receptor is being given by the binding affinity of that particular ligand with that of protein. So docking is being done and to evaluate the ligand receptor interactions in the way that may discriminate the experimentally observed mode from others and estimate the binding affinity. So to find out the binding affinity between the ligand and the protein, we do this docking studies. Components of docking involve like typically protein ligand docking software consists of two main components which generally work together. First one is search algorithm. It generates a large number of forces of the molecule in the binding site. Consider like I have like okay, consider this my hand and this is the protein molecule, and I have one small ligand. Consider this is the ligand. So over here, how can this particular ligand consider this is the binding site? This particular small molecule can bind in this particular way or this way, or it can stand in this way. So in whatever way, the generating the large number of forces of this chemical molecule is what we call it as searching the algorithm. So then next function what occurs in this docking studies is like scoring function. So in each of these fours, like the ligand has been bound to the protein molecule, what is the binding affinity or the score representing the strength of the binding? How well? If I keep it like this, it may, if the protein has been vibrating, it may fall off. Whereas if I keep it in a proper way or like in a proper force, the protein might not allow, even if it is vibrating, it might not allow the ligand to move away from the protein. So that would be the binding affinity or like the strength of the binding affinity. So that is the scoring function what is being analyzed in the docking studies. So two things, remember, search algorithm. In search algorithm, a number of forces of a molecule in the binding site is being analyzed or like is being generated. Second thing is like scoring function. Each of this force what is being generated by the search algorithm, the binding affinity is being calculated. So the best force with minimum by I mean like a good binding affinity will be considered over here. So scoring function includes like hydrogen bond interactions, electrostatic interactions, van der Waal interactions, hydrophobic interactions, pi-pi bondings, and so on. So coming to the application of molecular docking in modern drug discovery, search database and rank hits for the lead generation, calculate the differential binding of the ligand to two different molecular macromolecular receptors. Docking is also a widely used tool in predicting the protein-protein interactions when there are two or more proteins or two or more macromolecules. That knowledge of molecular associations helps us in understanding a variety of pathways taking place in the living system, and it reveals us possible pharmacological targets. It actually gives us an information about the mechanism of action too. Which particular protein does the ligand go and bind? When it binds, what is the activity which is being shown? So mechanism of action can be analyzed. It is less expensive than high throughput screening, faster than the conventional screening methods. So for conventional screening method is like you need to synthesize the compound, go for in vitro studies, go for in vivo studies. There a lot of time is required for that. So it is this particular method is very much faster, scanning a large number of potential drug molecules in very less time. So molecular docking studies over here can be done like even for a if you have a good system and all the softwares available with you, you know the methodology how to do, even you can do it. So it is so fast. So this is how the molecular docking studies come into use in the computational drug discovery. So, growing evidences of success, which are the compounds which are being synthesized or like which are being successful because of this particular computer-aided drug designing. Certain examples are like the discovery of indanavir, the HIV protease inhibitor. Then we have saquinavir, even that one is a HIV protease inhibitor target. Relenza, it is like neuraminidase is the target for that one. We have dorozolamide over here. Carbonic anhydrase is the target for that. Identification of haloperidol as a lead compound in the structure-based design for non-peptide inhibitor of HIV. So these are the certain examples what I have given over here as the success stories of CAD, computer-aided drug design. So what all we require prerequisite for molecular docking is like a valid hypothesis is required. Mainly, what is the problem? What is the problem? What exactly you require to do? That hypothesis is to be generated. Optimized protein structure is required. Optimized ligand structure or the structures whatever you have designed is required. Then various docking softwares. So as I have told you in the previous slides, like what few of the softwares is being told you. So Autodoc Vina is one of the free software. We have BioWire Discovery Studio. We have Schrodinger's and so on. Various free tools, web servers are free. Academic free tools are available, and commercial tools are also available. Then we need visualizing tools like PyMol or like BioWire Discovery Studio and so on over here. So these are the prerequisites for molecular docking studies. So various chemical ligand databases are there, where we can get the details, complete A to Z details of that particular ligands, that include impact, AromaDB, Dr. Dew's phytochemical or ethnobiological databases. So these are certain databases in which we will be getting the complete data of the ligands. ChemSpider, DrugBank, PubChem, TCM, ChemBay, all these are the various what I can say databases. Coming to the protein target databases, the major protein database is like the Protein Data Bank itself, PDB, from which generally we download the protein structure and so on for the docking studies. Binding MOAD, so mother of all the NR list. So these are the various what I can say like the protein target databases. So this is about the molecular docking studies. So next, let me go into a little bit about the QSAR studies. QSAR studies over here means like generating the theoretical models that relate quantitative measure of a chemical structure to a physical property or a biological activity. So generally over here we are relating the physico-chemical properties of a certain molecules with that of the biological activity. Physico-chemical properties are also been what I can say, like we can generate the quantitative measurements, or like we can give a mathematical equation or like a value to that of physico-chemical properties. So that mathematical values is being taken. Biological activities also can be given the mathematical values like IC50, ED50, and so on. So these physicochemical properties, the simple physicochemical properties is like molecular weight, molar refractivity, partition coefficient, so various lipophilic properties and so on, molar refractivity, and all these are the various physicochemical properties. I've just given you a few examples. So these physicochemical properties are being related with that of the biological activity. That means how does the biological activity chemical properties? If you have a certain list of compounds with different physicochemical properties and the biological activities, you derive an equation with this known information. So that means I'll just show you over here. There are various compounds like known activity or toxicity we are knowing. So consider you have 50 compounds of known activity. We calculate the physicochemical properties of all these 50 compounds, or like considering 40 compounds or like so as the training set and 10 compounds or so as the test set. We have the biological activities of all these compounds. So we relate like these are the biological activities which are required for the showing this particular activity or showing this particular toxicity, and we validate it by using the test set. After you validate the mathematical equation by using this particular way, then what we do over here is like unknown activity or unknown compounds are being taken. Unknown compounds, anyhow, the physicochemical properties can be calculated theoretically. So after calculating the physicochemical properties of those unknown compounds, you can predict their activity by the mathematical equation what you have generated. So this is how we can get to know like whether the molecule can show the activity or cannot show the activity. So that is what is this QSAR all about. So coming to this QSAR studies, the general workflow of the QSAR is like first thing is like generation of the molecular descriptor from chemical structure. Molecular descriptor is being generated from the chemical structures. After the descriptors are being generated, we have to screen these particular descriptors. Which descriptors are necessary, which descriptors are not required. We screen that, and only those descriptors which are prominent values are being considered in the test set. When you divide this particular components into training set and the test set, you see to it that it is like 40-60, or like a training set should be always more 7:3 ratio, or like 8:2 ratio, or such kind of ratios is to be taken. Training set is more and remaining at the test set. You are going to do the QSAR studies. QSAR studies means derive an equation, mathematical equation, relating the physicochemical properties of a drug molecule with that of its biological activity. After you get a proper mathematical equation, we need to do the model validation. Internal validation is being done, or external validation is being done. So over here, model validation over here means like whether the mathematical equation what you have got is good enough or not is being checked. So So over here, after you check that it is whether good enough or not by various ways, like leave one out method, leave many out method. There are various ways over here. Then we go for the model application. Model application is like after we design all these things and we go about with all these particular, we have got a mathematical equation. Then you will have to apply that particular knowledge or like whatever the mathematical equation is being generated to analyze the biological property of a new molecules. So this is being done. So later on, documentation of whatever you have done, publishing or getting a patent of that, or such things can be carried out over here. So this is the general QSAR workflow. So this is about the QSAR workflow. So the next is like finally coming to the key takeaways. So what all we have seen over here is like computational studies offers a compiling opportunity for the pharmaceutical organization from drug discovery and development. This computational studies, like whether it might be like from the docking till QSAR or like anything, statistical analysis and so on. Computational studies allows organizations to more efficiently manage and extend the product life cycle, increase the communications, which provides the patient, provide with providers, patients, meet the growing demands of the stakeholders. QSAR is mathematical relationship between biological activity and the measurable physicochemical properties for prediction of activity in silico. So these are the key takeaways of this particular presentation. So in this particular presentation, I have given an overview of the computational studies with a little brief introduction about the molecular docking and QSAR. It will take a lot of time for us to, what I can say, like explain this or like elaborate this particular topics. So now, if there are any questions, I'm ready to answer that.
Yes.
And
Thank you, Dr.
if there are
Mahesh.
any
It was
questions,
very
you can just take over it.
Yes, thank you, Dr. Mahesh. It was very valuable session. I'm sure our audience learned a lot from your session today. Let's take some of the questions we have. Like first question is it possible to do toxicity profiling of the candidates and how?
Ma'am, I could not hear you properly. Can you please repeat it again, or you can type it over here?
Yes, is it possible to do toxicity profiling of the candidates?
Okay. The question over here, your question over here is like, is it possible to find out with the toxicity studies also through QSAR studies? Was that the question?
Yes, yes, sir.
Yes, it is. It is possible to do the toxicity profiling also of a drug candidates by QSAR studies. It is possible. So by knowing like which particular physicochemical properties are responsible for the toxicity of a particular component, we can do the toxicity profiling also of the candidates, ma'am. Yes.
Okay. Adding to Dr. Mahesh, in Bioware Discovery Studio, we have a different Topcat protocol tool in our Discovery Bioware Studio where we can do the toxicity profiling.
Is there
We
any other
have
question?
different 12 models into it. Yes, the different, carcinogenic model, mutagenic model, even the aquatic models we have under Bioware Discovery Studio. Next question. What is the difference in pharmacophoric and QSAR models?
So coming to the pharmacophoric model over here, mainly we look into a part of the molecule which is responsible for showing the activity. Whereas QSAR model over here is like we have the physicochemical properties of all the molecules and we check out with the physicochemical properties of the whole molecule or a part of the molecule over here. And we do the toxicity profiling to check the biological activity. So that is the main difference over here between QSAR and pharmacophoric model, but it does not have much difference over here. That's what I can tell you.
Okay. The next question is it possible to do protein-protein docking using BioVia Discovery Studio?
I guess Sakshi Madam would be giving a better insight over this one because she's into that she's expertise in that, I guess.
Yeah, we have a protein-protein docking.
Protein docking
And okay.
via Discovery Studio. It is possible, man.
Yes, yes, definitely we can do the protein-protein docking using the BioWare Discovery Studio, where BioWare Discovery Studio works with the ZDog algorithm. Even we can refine those kind of scorings on the basis of RDoc refinement process. Even we can process those poses which we have got while doing these protein docking. So yes, definitely we can go with the protein docking using the software, where we can also use DNA and protein structure, RNA and protein structure, those kind of means those kind of conformations also we can check. The next question is, can we take the protein which does not show any binding site? How to identify those sites?
Okay, there is one more question over here.
Yes, can we take the
Can we
protein
take
which
protein which does not? So over here, Sudhir would not be showing any sites. We have various softwares or online softwares itself available, like CAST P and so on, to identify the binding sites. So that would be the answer for this. Can we take the protein which doesn't show binding sites?
is
Or you can
adding
go
to
for
Dr.
like flexible
Mahesh?
docking over here to identify which are the at least what are the binding sites available. So that can be done.
Next question is how to screen the molecular descriptors.
There is one more question like how to screen the molecular descriptors. So coming to how to screen the molecular. Hello,
Hello? Yes, sir. There is another question is how to screen the molecular descriptors.
madam, am I audible?
Yes, sir. You are audible.
Okay, how to screen molecular descriptors? Like we can go about with using the softwares like QSAR INS. So that is one software which I generally use to screen these molecular descriptors. Or molecular descriptors also can be screened manually. Like you need to know which are the descriptors, which descriptors are like which are responsible, or like there will be a number of descriptors. In that zero descriptors or the descriptors which have the same values, all those things, such kind of descriptors can be omitted, and most of by the softwares like QSAR INS. Or if you have enough time and you know the have the knowledge of the molecular descriptors, you can do it manually also.
Okay, thank you, Dr. Mahesh. I think we have done with our questions for the session. Uh, it was very great presentations and very knowledgeable too. It was our pleasure
Okay,
to have you today here with us.
okay.
And uh, yes, thank you all. You can
Okay,
subscribe us to
I
stay
need
tuned
a
with
minute
us.
more.
Yes,
Is it
yeah,
possible?
yeah, sure, definitely. Yes, yes, sir.
Hello.
Please. Yes, sir, definitely.
So before the session has been ended, I would like to
Yeah.
just inform we are organizing a new international e-conference in the on 28th July, which the link will be shared to you people. So if you're interested, you can just go ahead with registering with this one. And then I would like to acknowledge all the participants. I would like to acknowledge my institution, MS Ramaya University of Applied Sciences. I would like to acknowledge Dr. Parshuram P for helping me out in preparing these slides. I also thank Dr. Sakshi for inviting me to present this particular presentation. Thank you, ma'am. Thank you very much.
Thank you, sir. Thank you so much. Thank you all for joining us. Thank you.